welcome back to Now I Know. Today let's see difference between genome, chromosome, gene and DNA. Now these terms, even though we use it very frequently, I have seen students getting confused between them. Even when I was studying in my plus 2, I used to get really confused between chromosome and DNA especially. So today in this video we are going to clear all our doubts regarding these terms. So let's begin. Alright, so we'll start with DNA because all of us will know the basic structure of DNA. It's a double helix structure. It has a sugar phosphate backbone in it, and it is made up of 4 nucleotides: adenine, thymine, guanine and cytosine. Adenine pairs with thymine, guanine pairs with cytosine. Alright, now one important point that we should know here is when you take one eukaryotic cell, now we are talking everything in terms of human, okay? So when you take DNA from one nucleus, one cell, the nuclear DNA would be around 2 meters long. Now we have talked about this in DNA packaging video. I'll put the link on the screen in case you want to see that this huge amount of DNA needs to be fit inside tiny nucleus, maybe around 5 to 10 micron in size. So it has to undergo packaging strategy, and we have seen how the nucleosome is formed and how it is arranged in chromosome. Okay, but still keep this point, this 2 meter long DNA, just pin it over here. We'll come back to this. This is very important to understand. So DNA is double helix, has sugar phosphate backbone and made up of 4 nucleotides. So this is simple to understand. We are done with DNA. We are clear about DNA. Alright, now after DNA comes genes. I'm going in a flow, okay, that will become easy to understand. Now as I just said, the DNA in our nucleus would be around 2 meters long. So this long DNA, in this long DNA, the segment or chunk of DNA, that means when you say segment or chunk of DNA, now we know it is made up of what? Nucleotides, A, T, G, C, all different combination will be there. In that, one specific sequence of nucleotide, maybe some 100 base pairs long. Okay? That specific nucleotide sequence, that chunk of DNA is called gene. Okay? There are specific nucleotide sequence or segments or chunks of DNA which can be some 100 base pair long. That is gene. Okay? That is a specific sequence which would code for specific protein and RNA. We know that everything will start at this particular point. DNA has to be transcribed and translated for functioning of the organism. So this specific sequence, which can be some hundreds of base pair long, in the other way we will say segment or chunk of DNA, that codes for specific protein and RNA is called gene. Thousands of genes throughout the DNA. Don't look at this term as of now again, otherwise you'll get confused. So we'll have about, in humans, about 20,000 genes are there in our DNA, the whole of DNA, complete DNA. So now we might have heard about, the traits or specific characteristics that we get from our parents, that is because of the genes. That is because these genes gets passed on from one generation to other. So genes are nothing but specific chunk or specific sequence of nucleotides that codes for protein and RNA. And these genes are generally few hundreds of base pair long. Here I have shown only few, but in reality it is actually some hundreds base pair long. And one example that I have given here is cystic fibrosis gene is present on chromosome 7. That means every gene has a specific site in our DNA. Where it is located is very specific, and it will be the same throughout the population. Okay, now from gene, let's move on and take a bigger picture. Alright, so we just saw that we have really huge DNA, 2 meters of DNA to fit inside the nucleus. So for that, if you try to, put this DNA as it is in such a tiny nucleus, it's not going to happen. So nature has a beautiful mechanism of packaging of this DNA into nucleus, and we have talked about that mechanism. We are not going in detail, but just briefly, if you look at here, DNA we know is a double helix. Now we know that, and genes means specific nucleotide sequence that is present in DNA. Now this thread, okay, DNA is like a thread, will start wrapping itself around the histone proteins. All right. Now, when it does so, what happens? It results in reduction of its size, as in the space that it might require to fit in would reduce. Okay, so it starts undergoing condensation process by wrapping itself around the histone protein, and that results in formation of nucleosome structure. As I said, I'll put the link on the screen for this mechanism, condensation and formation of chromosome. In detail, you can have a look there. It will further undergo coiling. There will be arrangement of these nucleosomes. So it's like histones are there everywhere with DNA wrapped around it. So as we can see here, the nucleosomes are getting arranged, is getting more and more condensed, resulting in this particular structure over here, which we called chromosome. Alright, we are familiar with this structure. This is nothing but super condensed form of DNA where it is wrapped around histone proteins. Alright, now in humans we have how many chromosomes? We have 46 chromosomes. Because we are diploid organism, what happens is we have one set of chromosome that comes from mother and one set of chromosome comes from father. So that means we have 23 pairs of chromosome. Now, out of this 23 pairs, we'll have 22 of autosomal chromosomes and one pair of 6 chromosomes, which can be either XX or XY, right? Now, I told you just pin this 2 meters point over here. Now let's take that 2 meters of DNA point and try to see here. We say it's about 2 meters long. The DNA is about 2 meters long. When I say the DNA is about 2 meters long in one nucleus, what I mean is when we take all of these chromosomes, all the 46 chromosomes, and unwind the DNA and put them all in one long strand, the whole length of those 46 chromosomes in one cell is about 2 meters long. Alright, that's what it means. Now this is one point where there can be confusion. Okay, when you say DNA is about 2 meters long, we are talking about the complete DNA, that is all of the 46 chromosomes together make up the 2 meters DNA, and that has to fit inside our nucleus. When it comes to gametes, they are going to be haploid. Right, so they will have only 22 of autosomal chromosome, not pairs. Only 22 of autosomal chromosomes and one of the sex chromosome, either X or Y. So this we are talking about somatic cell, a diploid cell, 46 chromosome, 2 meters of DNA. Keep this point very clear in your mind. Alright, so now we are done with DNA, gene and chromosome, right? So what is this genome then? Alright, so it's easy to understand. A genome is nothing but an organism's complete set of DNA. Okay, what is this complete set of DNA? That means we are talking about humans. So we have all the chromosomes present, all the DNA that is present in nucleus. Other than that, we also have DNA present in mitochondria for us, right? Say for example in plants, if we are talking about, we'll also take in consideration chloroplast. So for human, the complete set of DNA, that means you need to consider all the DNA that is present in nucleus. That together makes up the genome of an organism. It's nothing but you are taking in consideration all the genetic information that is present inside a cell of organism. And inside a cell of organism for humans, we have nuclear DNA and we have mitochondrial DNA. So when you put all of that together, it becomes the genome of an individual. Now to make this very simple to understand and just put it in one flow, let's look at it this way. In one cell, if we take, we have a lot of genome, right? A lot of information is there for one cell. Okay, now imagine this information is nothing but it's in form of nucleotides, right? There are specific codes, specific sequence of nucleotides that is going to tell the cell how to grow, how to survive, how to develop and so on. So there is a lot of information in form of genome. Now imagine if this genome, let's imagine this as a huge textbook that you are studying. So it's a huge amount of data containing one book. If you don't have chapters in the book, what is going to happen? It's just a lot of information written all together. That is going to be little difficult for you to go through it or to access it whenever you want. So what nature did is this genome, which has a lot of information, nature divided that into pieces of chromosome in 23 pairs of chromosome, right? So that becomes easy. Now this whole lot of genome, which has a lot of information, is divided into 23 chapters, say for example in books. Now each chapter is going to have different information in it, so you can access it whenever you want based on what is your need. Particular chapter you can go through. Similarly, all the genes that are present in the genome are divided now among these chromosomes. So it becomes very easy for cell. There is a specific site for each chromosome. As I said, say for example over here, cystic fibrosis gene is present on chromosome number 7. A specific site will be there, specific locus will be there. So genome is divided amongst these chromosome, right? Again, when you take chromosome into consideration, you will have specific chunks or segments which are coding for specific proteins that are nothing but genes, and the basic unit of all this is your DNA. That's all. This is very easy to understand, right? You can go either way. From DNA you can go to genome or genome to DNA. So that's all. That is what is DNA.